Good afternoon, everyone. Kind of weird to be back here again. Hopefully, the stint online wasn't too awful for everyone. Um, Hopefully, and you saw some of the announcements and stuff that have been going out. I'm in the process of drafting another announcement. I wasn't quite sure how long it would take to get through the door this morning, so I came early, which cut into my announcement putting together time. But I'll be posting this on Moodle as soon as I'm done teaching today. But this is basically letting you know that we finally have access to all of the midterm marks. Those have been released to you. You might have seen them already. I have done enough versions of this class that I have a class average that I usually expect to see, which is about a 70%. And we didn't quite hit that mark. And I think it had something to do with this sudden move to online and everything being very different. So I'm making two adjustments to our grades just to try and help with something like that. I really hate when we don't get a representative measure of people's marks. So the first thing I'm doing is kind of an easy one, which is I'm reducing the number of questions that it's out of. There weren't any questions that everyone did poorly on, but I basically took the highest grades in class and divided everybody else's grade by that instead of, it was out of 45, so I think I went out of 43. So it looks like it works out to be about a 3% straight boost for everyone. So that's the quick and easy one. Um, the other option that I'm doing is because we had such a dramatic swing in how the midterm was delivered from how we planned, I'm actually going to do a sort of a secondary way of distributing the grades. I'm going to calculate the original one that's in the syllabus. I'm going to calculate a second version where I just weight that first midterm as less. So for right now, we have sort of 25, 25, and 30% for the two midterms and final. But for those who didn't do super well on the first midterm, I'm going to drop that one down to, what did I say here, 15? Um, so 15 and then 30 and 35 for the second and the final. So I have that written down. So we'll just take the 10% off of the weight of the first midterm and scoot it to the other two. And that way, so if you ended up not doing as well as you would hope on that midterm, it's not going to affect your grade as dramatically. And then hopefully the next two will be in person as planned, as opposed to thrown together in the last week before the exam. But hopefully that makes sense to you guys. And like I said, I'm going to be putting this announcement up on eClass so that you can actually see it. Um, I haven't found a way to make Moodle show the new total. If I tell it that it's now out of 34, it then scales your grade, which doesn't help anyone. So it right now shows out of 45, but this announcement is telling you I'm treating it as 34. Or I'm saying 43. Thank you. I'm so sorry. I don't do well with numbers if you haven't figured that out yet. Um, Anyways, yes. So all the numbers that are written down are correct. I apologize for the verbal swap, which happens very often. So as I said, yeah, at the end of the semester, I'll calculate your grade both ways, the original and then that adjusted one, and I'll just take the best of the two. So it's not going to negatively impact your grade, it's only ever going to make it better. So that's how I make adjustments if things have gone a little bit awry. But it was such a strange thing to look at the distribution. Like I said, I've done quite a few sections of this course. And there were some people who did phenomenal, had almost marks, and then there were some people who it just didn't click for them. And that's why I think it's the method of delivery. So making some adjustments, and I just wanted to let you guys know about that now that the grades are available. And if you've taken a look at that, you might have noticed that you also have access to the full exam. So you can go in and look at your questions, look at the answers, and especially for the written questions, if there were any grader comments, if I thought anything you said, I made a little comment. And that's because I usually do like in-person exam viewings, but that's really hard to do right now, especially where us instructors don't have offices. So that's the way we're going. If you have any questions, the usual, just take a screenshot and send me an email. If you want to go over more than one thing, we can absolutely set up a meeting to go over stuff, whatever works for you guys. But that's a lot of rambly admin stuff at the beginning. Last piece of rambly admin stuff is I'm still recording our lectures here in the background. So if you're finding it's a hassle to come to campus, or if you're finding it's difficult or you just don't want to be here, that's totally fine. Everything's recorded and I'll post it. I usually set it up so that it's processing on YouTube while I run to my next class. So sometimes it's up very, very quickly. But yeah, just giving people some options. So into actual lecture topics now. We're going to start talking about the next of our two systems. And I kind of mentioned in our recordings that I'm doing these a little bit out of order. The textbooks always seem to start in vision, and I'm like, I don't know why they're starting with the most complex system. I like to build our way up to vision. I like to establish the terminology. I like to get a feel for what we're talking about and how we talk about it, and then get into vision. So we're going to end on vision. That should fill next lecture entirely. Um, Hopefully not much more than next lecture because I'd like to move on to our next chapter, but we'll see how it goes. But today we're going to talk about our chemical senses, so olfactory and gustatory systems. And then I'm going to also talk about some of our sort of physical touch sensations and our kinesthetic and vestibular systems. And in comparison to both the auditory system and the visual systems, each of these is kind of like a micro lesson. They're a lot smaller, a lot more precise. And sometimes it's because we don't need tons of detail, but I can also let you in on a little secret, and that's the fact that we don't actually know a lot of detail about how some of these work. So when they say that we're still doing research, it means they have no clue, but they're going to try and find out, which I found very, very interesting. All right, so starting off with olfaction or our sense of smell, you're going to find that smell and then the next topic, taste, are very closely associated, almost intertwined with each other. They both end up using physical sort of chemicals that are in the air or when we get into taste in our saliva in order to sort of turn that into some kind of meaning. So you'll sometimes see this described as odorants, things that have smell. That just means substances that are carried in the air. One of my favorite definitions that I saw from a textbook, let's see if I had included it in here. Oh, I didn't. Oh, well, that's unfortunate. It was something like volatile chemical substances, which is accurate if you look at it chemically. Volatile just meaning they're aerosolized at room temperature. But the way that it was worded, it sounded like it was something very aggressive and intense. But if we think about it, things that we can smell are just things that have sort of evaporated and are carried in the air. So these are things that get into our noses, into our sinus passages, and then they dissolve in the fluid, the mucus that's in our nose. So the same way when we talk about taste, we'll talk about those smells being dissolved in our saliva. Same thing with our nose. And that's why there's so much crossover here because taste and smell end up being very closely interpreted, and the two signals get put together in the brain in a lot of cases. So here are our physical stimuli are those odorants, those chemicals carried in the air, and the receptors are our olfactory cilia. So if we look at the next slide, there are so many different images of this, and none of them are super helpful. But basically, we're looking inside that open cavity in our brains. If you've ever had a sinus infection, you know how awful it is to get that burning feeling sort of above and behind your mouth. That's that big open passage. And so along that in the lining, we have those olfactory cilia. So they're shown here as blue, sort of spreading out nerves, or we'll call them cells. So those are our receptors, and they feed back into our olfactory bulb. And that olfactory bulb is sitting right at the base of our brain. So it doesn't even have to go through all of our brainstem and stuff to get into the brain, right at the base there, and get distributed pretty much internally. Um, and for our pathway explicitly stated, so we have those olfactory cilia, and that's where that transduction happens, where the sensation, what's being brought in, is turned into a neural stimulus, and then it's going to go along those olfactory nerves into the bulb at the base of the brain. And this is the only smell that or the only sense that we spend a lot of time talking about that completely bypasses the thalamus. And when we talked about the thalamus, we talked about how important it is to sort of spread out and redirect all of our sensory information. It's sort of our routing station. But our sense of smell is considered one of those very basal abilities where it would have evolved so far back that it might have happened before we had as complex of a structure as a thalamus, which is why it wasn't really needed. And the concept of smell is a pretty basic way of interacting with our environment. It is fairly unfortunate that humans are very bad at smelling. Um, so it's not that we don't have the ability to perceive all the smells that everyone else can. It's that we need higher concentrations of those smells to be able to smell things. So we are very insensitive to smell, is probably the best way to say it. So if you look at something like different species of dogs, some of them have, I think it's as high as like 40,000 times more sensitive smell, which is phenomenal. All right. And then the next thing I wanted to point out, like basically all of our senses, the sense of smell shows sensory adaptation. So if you've ever seen those Febreze commercials where they talk about being nose blind, that's sensory adaptation, where at first a smell is really, really strong, but over time you notice it less and less. So we can adapt to that because we don't have to pay attention to it because it's constant. So from an evolutionary point of view, it makes sense where if you're living in a forest and it always smells like pine trees, then it isn't useful to know that it smells like pine trees because it never changes. But if all of a sudden it smells like a new kind of tree, that might be important. Um, and they talk about our perception of an odor usually fades by about half its original strength or more within 4 minutes. So you walk into a place, you notice the smell really, really strongly, and within 4 minutes, you almost don't notice it at all. And when we look at perceptibility when it comes to smell, they seem to find that females are a little bit more accurate at categorizing smells than males. I find this interesting because I have almost no sense of smell and have for a really long time, which was terrifying when that was one of those symptoms of COVID because since I was a child, I have like four smells that I can smell and that's about it. So when we talk about stuff like this, I was like, well, I'd be an outlier. But yeah, there's lots of interesting studies that look at are we better or worse, or who's good at picking up which smells. And then we have some smells that we are more biologically predisposed to pick. So if you've ever heard of pheromones, those are chemical messages, and we're not usually consciously aware of them, but they can influence our behavior. So, what's our? Oh, okay. Um, So with humans, we can smell each other's pheromones even if we don't know that we can, and even if we're not aware of the fact that we are. And there's some really neat studies. This is just sort of an aside because it's neat. Some of the studies that I've seen, they do things like having a bunch of male university students who go out and play basketball, and they're giving them like a specific shirt, a white cotton jersey, and they say, "Go play basketball for an hour, then give us your shirt." And they will get other people to come in, smell the shirts, and rate them on attractiveness. They will then also have pictures of the people who wore those shirts and get other people to rate them on attractiveness. And there's a really strong correlation between the two scores, which just blows my mind. But apparently, they've done it multiple times in different populations, and it always correlates very strongly. So that's your, I guess, gross science fact for the day. And then that's probably a good enough place to move on to our sense of taste. As I said, it's very choppy here, where we're doing just sort of the highlights of each of these senses. So when we talk about taste or gustation, which I always pronounce with a hard G, even though I probably shouldn't, um, this is where we have again our physical stimulus is going to be soluble chemical substances, so things that we can dissolve in our saliva. And our receptor cells here are found in our taste buds. So this is one of those weird things where when you think of taste buds, you think of that sort of bump on your tongue and you assume that the peak of the bump, that's the receptor. But in fact, it's in the grooves between taste buds. So in those valleys between the bumps, that's where our receptors are. So those taste receptors are along the edges of the bumps that we think of as our taste buds. And you might have seen, especially in elementary school, they like to do this map of the tongue where they show that you taste bitter at the back and sour and sweet, uh, or sour and salty at the sides and sweet at the front. And that's not really accurate because that gives you the idea that you can only taste bitter at the back and nowhere else. And you can't taste any of those other flavors at the back of your tongue. And what we have instead is sort of a distribution where different parts of the tongue will have different concentrations of those different receptors, but not really mutually exclusive. They're all found kind of everywhere. And this is one of those things where we have very good agreement on the four tastes, the sweet, salty, sour, and bitter. But modern research is starting to show a lot of evidence for a fifth, which is umami, and that's sort of a savory flavor. So some texts say that it's there, some texts don't, some like mention it but don't group it in with the four. I like including it because I do think with the amount of research coming out on it, it's probably going to be in all the texts eventually. And that's one of those first things where I'm going to watch it change over in all my books. But um, for that idea of where the taste receptors are. So the taste buds, those receptors are in the grooves in between the bumps. So the bumps themselves are not the taste buds, they're in between those bumps. And then if we look at that graph there, you'll see that it shows four different kinds of receptors. Receptors for sweet and sour or salty and bitter, which I can't say in the same order. And they show different concentrations of those receptors at different places along the tongue. So at the back of the tongue, yes, there are more bitter receptors there. So it's easier to taste bitter at the back of the tongue, but you still have salty, sweet, and sour receptors there too. So you don't just taste bitter. That's just the strongest thing to pick up there. And same with sort of the middle and the front, we have that different distribution. So that's why they kind of called it a hard line between where you can see each of those things, but it's a lot more sort of varied, I guess we can say. And then if we just wanted to look a little bit more in detail about each of those taste buds, so inside that groove, when we have each of those individual cells, it's not one cell on its own, they tend to be grouped together with sort of 50 to 100 different taste sensitive cells. And this is another one of those where our understanding of this isn't fantastic, which you might have gathered from the fact that we're introducing a new fifth taste. So this is gradually advancing as we look at it here. But this is just showing that it's the same basic setup as all of the other systems that we've looked at, where these cells sort of feed back. There are neurons that feed that information back into the body so that we can perceive whatever sensations are coming in. And then again, for our explicitly stated pathway, we have our taste buds, and that's where that transduction happens, going to convert that sort of binding of molecules into a neural impulse. This one goes through the thalamus and then up into the cortex. So, like normal senses, we're going back through the thalamus and then into the cortex. And we have sort of a really interesting combination here where our sense of smell and taste both get combined in our orbital frontal cortex, which, if you don't want to write it down, is on the next slide. But they're going to combine and come together. Yep. Yeah, probably. I think so. I'm sure there's a lot of debate on that, but all we can do is sort of look at common ancestors and see how far back each of these traits have evolved. But the assumption here is, yeah, smell first. It's very cool. Um, and we do have, just talking about taste alone before we bring it back in with smell, some of our basic taste preferences may be innate. So there is some evidence for us enjoying certain tastes from a very, very young age. So you can think of small children absolutely love sugar, sweet things. And that kind of makes sense evolutionarily because a lot of things that are very sweet tend to be very high calorie. And that's problematic nowadays when it's easy to get access to lots of high calorie foods. But if you think of us living sort of out in nature, you'd want, as a very young child, to eat a lot of high calorie things so that you would take in enough nutrients and grow. So it was helpful prior to society now, but now it's causing some problems. Um, so there's that for basic taste preference. And I've also heard, I don't think the textbook talks about this, but again, studies of interest. As you get older, your preference for sweet drops off. I don't know about you guys, but when I was younger, I would eat milk chocolate from sun up to sundown, no problems ever. And I like would try dark chocolate because it's healthier, and it was awful. And it wasn't until I was like 25 that I could tolerate dark chocolate. So I don't know if it's a genetic, like as I've gotten older and developed, or if that's just I've been exposed to it, but there is some evidence that our preference for sweet goes away as we get older, which is kind of unfortunate. Um, uh, yeah, so that's the physiological mechanisms that might be controlling. We also have a little aside that we can talk about. Not everybody tastes the same way. So there are some people who have like sort of a standard amount of taste ability. There are some who are on the lower end of that scale, so they are non-tasters. And then you have people on the other end of the scale who are super tasters. And so those people who have sort of either uh, higher concentrations or even just a better awareness of the receptors that they do have, they tend to be a lot better at picking out sort of individual flavors in a dish. Maybe you have a friend like I do who can have any dish no matter how complex and is like, oh yeah, I know exactly what spices are in this. As someone with almost no sense of smell, I'm just like, cool, it tastes sweet. But that would mean that I'm on that sort of non-taster end of this. But we do tend to have sort of a wide variety. And these variations in how people taste food actually leads to differences in our behaviors around food. So people who are super tasters are a lot more likely to eat healthy because they're aware of what they're eating, and so they're going to make better meals. And a lot of times they'll choose to make their own meals because they can control each of the ingredients that are put in, which is kind of cool. And then there are the people who don't have a fantastic sense of taste and might just eat Kraft dinner every time they get the chance. Um, and then I guess, yeah, our last thing is that most of our taste preferences are learned. So we do have that innate, love of sweet when we're younger that sort of tapers off as we get older. But most of our preferences for taste come from exposure. So if you grew up, as I did, on meat and potatoes and that was it, then that's probably what you grew up enjoying. So maybe you hadn't been exposed to other things. Maybe you didn't eat rice until you were older. So maybe you're not a huge fan of rice or something like that. So there's sort of cultural differences based on what dishes you've been exposed to where you were born, and also just sort of what your family has. So I know my mother doesn't like turnip, and so I never had turnip, and so I don't like it either. Not because it's bad, but just from lack of exposure. So there's stuff like that that we want to keep in mind where individual differences can affect the preferences as well. All right, and then with the combination, so when we think of taste, it often gets confounded with the idea of flavor. And flavor is sort of our technical term here for the combination of taste and smell, as well as a little bit of the tactile sensation of food in your mouth. So when we taste and smell things, you kind of need both senses coming together to get the full picture. So if you've ever had a cold and you've been really stuffy and then you find food that's usually very flavorful tastes bland, and that's because you're missing out on the scent portion of that flavor. So these are all kind of combined, and as I said, they combine in that orbitofrontal cortex. Um, so odors are a massive part of perception of flavor, which is probably why I would group myself on that lower end of ability to taste, is lack of ability to smell. All right. And so, as I said, that's it for those two senses, which is good, nice and quick. And if we think those are quick, we have three more that we're going to talk about. One is our sense of touch, which will be a decent length, and then we're going to do our kinesthetic and vestibular systems, which I think have like maybe 1 or 2 slides each. So it's just sort of a very, very quick tour of the body here. So starting with our sense of touch, we're going to be focusing in on the sensory part in our skin. So our skin is all around our body, and that's how we interact with our environment, and it has lots of different kinds of receptors. And this is, as I said, another one of those where there's a little bit of debate on how we classify things. What I find is that texts will say that human skin has at least 6 types of sensory receptors, which means that there are 6 that they can agree on. We talk about 4 of them, and we call it good. If this is something you're interested in, there's tons of research on this, and it's another one of those cutting edge. We're learning how all of these senses work together. But we're going to focus in on the fact that our skin gives us information about tactile qualities, so things that we touch, the sort of feeling of things pressing on our skin, and our ability to localize where that tactile sensation is coming in. We also have 2 temperature sensors, one that does good with warm and one that does good with cold. And those are going to let us know if we are hot or cold. And we also have pain receptors, so how we perceive different feelings of pain. And we'll talk about each of those individually. But overall, when we're looking at our physical stimuli, these are going to be either mechanical, thermal, or chemical energy that is contacting or impinging upon our skin. So if we think of our tactile quality, that would be something touching our skin. Warm or cold would be the chemical energy. Is there sort of lots of heat, lots of vibration in the molecules, or is there very little? And mechanical could also just be like if you get cut or something like that, that would trigger a sensation of pain. So lots of different kinds of physical stimuli here. And for our general pathway, we have our different kinds of sensory receptors. Those are all embedded in our skin somehow. And that's going to, again, have that transduction part here. I didn't include transduction here for whatever reason. I don't know why, but it's going to go into our spinal column, up our spinal column into our brainstem. There's a crossing over where we talked about earlier on, where the right hemisphere controls the left side of our body and the left hemisphere controls the right side of our body. That crosses over sort of as we've passed through the brainstem, and then our thalamus will reroute everything up to our somatosensory cortex. So that's in our parietal lobe, if you remember. So, and as I said, we have all of these different kinds of receptors that are going to then be sending information along that same pathway so that we know what each signal means. So this particular neuron is a pain receptor, or it's carrying the signal from a pain receptor. And this particular neuron is from a tactile receptor, and so on. So let's look at each of these then. We'll start off with pressure. So specific cells in our skin are going to be sensitive to touch or sort of that mechanical pushing on different parts of our skin. And so we have receptors that basically pick up on different sized parts of our skin. And so these are going to have sort of a predictable arrangement where our brain knows what each particular neuron is reporting from. So this part of my arm is different from that part of my arm, which is different from this part. And we can interpret what each of those mean in order to localize where we're being touched. So we have sort of an excitatory center. So the point where we're receiving contact, the most contact, that's where we're going to get excitatory signals. And immediately surrounding that, everything that's around it that's sort of affected, that there's a little bit of movement but not a lot, it actually acts opposite and it will be inhibitory. So we kind of get like a bullseye that says this specific point is exactly where you are being touched and everywhere else is not where you're being touched. So that's how if a mosquito lands on your arm, you immediately know where it is. It's because we have this ability to localize it very, very precisely. There are a couple of places, I don't know how or why, that can screw this up. The inside of your arm is one of those places where it gets a little bit muddy. I don't know if as children you ever did the thing where you like sort of draw little circles up your arm and try and get the stop your elbow, and you can be off by like 5 to 10 centimeters. And it's just because that part has larger patches that it's sensitive to, so it's a lot harder to localize. But on the outside, we have very good localization. Um, yeah, so that lets us localize sensation, and we are very, very good at that on most parts of our body. And again, because it's going up into that somatosensory cortex, the parts of our body that are more important, that need to be more dexterous, things like our hands and our feet, might have more precise resolution than larger areas. And again, also subject to sensory adaptation. So when you put on a new shirt in the morning, it might feel a little bit scratchy, but by the time you're ready to leave the house, you don't even think about it ever again until your instructor reminds you about it, and now you're uncomfortable in your skin. But that's sensory adaptation for touch, where again, it's not beneficial to still be paying attention to things that are constant. It doesn't matter that you have this feeling of the way that your hair is today, because it's not helpful. But if something lands on the side of your neck, then maybe it's a fly, maybe it's a mosquito, we should deal with that. So we pay a lot more attention to things that are changing, things that are new, than to things that are constant. All right. Next, we can talk very, very briefly about temperature. Again, we have two different kinds of temperature sensors, one that focuses on hot and one that focuses on cold. All we care about is the fact that both are free nerve endings in the skin. So basically, our the neurons are going to sort of be spread out, they're going to connect back to that nerve, and they don't have a specific receptor itself, but the ends of our neurons are what are reacting. So it's not like a taste bud has a specific place for binding to something. These are just the free ends of our neurons that are reacting. And as you can probably imagine, this can be a lot more complicated than what we're going to get into for this class, but we'll keep it at that brief statement. When it comes to pain, we're going to talk just a little bit more about this. Again, are free nerve endings, so they are the sort of open ends of our neurons that are going to be able to pick up sensation. But it's a little bit more complicated than that because they feed into two different pathways. So we have a fast pathway and a slow pathway. I can actually show a visual of this. So we have our fast pathway, which is, I believe, oh, I didn't label them on this. Hold on, let me scroll so I don't say it backwards. Slow is the C fiber, fast is the A delta. I always say that. Okay, so the red is the fast and the blue is the slow. So that's just sort of for your reference. I'm not going to test you on the specifics of that. I just like seeing it. But basically what's happening here is that with our fast pathway, they're going to register the localized pain and send it up to our cortex as fast as possible. So those neurons are all myelinated. They're going to send that signal in fractions of a second so that our body can immediately react. Our slow pathway is going to be less specialized, less fast. It's going to give us that overall feeling of this hurts continually. So the fast thing is going to be a localized, precise, it hurts right here in this way. And then the slow pathway, those are non-myelinated neurons. So they're going to travel a little bit slower, but they're also going to give a feeling of sensation for longer. So when you stub your toe and you have that very, very sharp, immediate feeling, and you have just enough time to go, oh no, this is going to hurt, and then it starts aching, that's the difference between your fast and your slow pathways, where one of them happens pretty much the second your toe hits the edge of that coffee table, and the other one is going to hurt for longer and almost feel a little bit more intense in the long run. But that's just because it's taken its time to get there. So those are our two different pathways. And as I said, there's a lot more detail we could go into, but we keep it a little bit brief. And I want to check, that clock is still broken, which makes it really hard for me to judge our timing here, but I think we're doing good. So, okay, that's how that pain pathway works. That's the two different ways it goes. But how do we actually feel pain? How do we perceive it? And there are a bunch of really cool things that are involved here. And again, lots and lots of research that could be discussed. But the perception of pain is influenced by a ton of different factors. It is the perfect thing to talk about in psychology because things like our expectations, our individual personality, our current mood, all sorts of different factors can affect how we experience pain. So even though the sensations might be similar, the experience, the perception can change based on an individual and their current state, which is really, really interesting. So things like looking at pleasant photos or seeing a loved one can actually make you feel pain a lot less acutely. So by having something nice around makes it a lot less bad. So if you're having an awful day and you've stubbed your toe, if you look at a picture of a cat, if you like cats, that might help a little bit. Um, we also have some interesting studies that show that an individual's intention, so if you are intentionally caused harm, so somebody comes up and pokes you with a needle, you perceive that as being a lot worse than if they accidentally did it. I'm not sure how they would accidentally poke you with a needle, but we can see. So they've done a bunch of different studies on intentional pain and unintentional pain, and people rated the intentional pain as a lot worse than unintentional. With that expectation idea that might make you think back to the placebo effect, where your expectation of what should happen actually influences your perception of that event. And sure enough, it does. If someone is given a sugar pill and is told that it's a painkiller, you give somebody a mint and tell them that it's an Advil, their headache will reduce in intensity. Their perception of that pain will get less, which is really, really neat. And then there's also a lot of cultural differences in pain perception. And this is one of those interesting fields where um, there's been lots and lots of studies, and some of them have been framed as it's a difference based on ethnicity, it's a difference based on location, it's a difference based on whatever. But most of the studies, especially in psychology, have focused in on it's not so much an actual difference in their perception, but actually a difference in sort of what they're able to tolerate and how they frame the pain that they're experiencing. So, in a culture where you are used to being sort of surrounded by individuals and then you're alone, that loneliness may be a lot more painful than if you came from a culture that was a lot more used to being on your own. So, it's not so much a difference in pain perception on paper, it's more of a framing of that perception. And of course, we can't talk about pain without talking about some of the possible pathways that help us control how we feel pain. So we get those sensations, but sometimes the feeling of pain ends up going away. We can talk about the idea of pain being adaptive, where it can be beneficial. If you have a blister on the back of your foot, maybe it makes sense to know that you should adjust how you're walking so it doesn't keep causing you pain. Or maybe you have a headache because you haven't drank enough water. So that's a signal to go drink water. So pain can be adaptive, but it's always only up to a point. If you're in a bad situation, say you're getting attacked by a bear, yeah, you got bit by a bear and the bite hurts, but that's not helping you fight off a bear attack. So after a little while, you will drown out that sensation and focus on something else. This is where our theories of sort of blocking out pain come into effect. And this is another one where there's a bunch of different theories, and it seems to be a little bit of a conglomeration of all of them, but no one will agree on exactly. The earliest of our main theories is gate control theory. And this is basically saying that our incoming pain sensations, when they're routing up through, say, our spinal cord, they pass through a gate. Not a physical gate, but sort of a pattern of firing type gate that our body can sort of turn off to close off and prevent those signals from reaching up to the brain. Um, I found this a really weird theory. I envisioned like a physical gate where you sort of block off those neurons firing up the spinal cord. But it's not really that. They call it an abstract sort of concept of a gate where the pattern of firing, if it doesn't sort of meet a certain pattern, they'll block it out. They don't go into much more detail than that because it is one of those weird old theories. I think it came around in the 60s. So this one, a theoretical, not physical gate that prevents those signals from being passed upward. The ones that make a little bit more sense for us are going to be the presence of endorphins, which I think you might have seen endorphins discussed very, very briefly in chapter 3 when we were talking about neurotransmitters and stuff like that. Endorphins are, they effectively mimic things like morphine and they provide pain relief. And we can find them all throughout our central nervous system. And having higher concentrations of these endorphins means that we perceive less pain. So that might be involved. And we have a little bit more evidence that that might be involved because of our next thing, which is the descending neural pathway. And this is a pathway that's sort of sent from our brain down to the rest of our body, and it mediates how we suppress pain. And it seems to be triggered by the presence of endorphins. So again, they're not positive of the mechanism there, but our brain realizes that we're in too much pain to continue functioning. So it sends a signal to basically inhibit those neurons that would transmit incoming signals, which is really neat. And again, of course, there's so much we could talk about here, but we're going to keep it moving because we are getting awfully close to the end of time. So for our last part here, we're going to talk about our kinesthetic and vestibular senses. And as promised, I think there are only 4 slides here, which is very nice. But our kinesthetic system ends up monitoring the position of all of the different parts of our body. So we have receptors in our joints that basically keep track of how much are your joints bent. So if we look at our elbow, how much is your elbow bent? Is your arm straight or inward? Um, how much is it moving or is it stationary? Those sorts of things. So it keeps track of that information. And most of that kinesthetic information is going to get transmitted to the brain along the same pathway as our tactile sensation because it's just receptors telling us how things are located. So it's going to tell us if the muscles are taut or extended, if our joints are bent, that sort of thing. So it's just a body position information. And then our vestibular system is in response to gravity. So not just what position are your limbs all in, it's are you upright? Are you laying down? Are you moving? Are you stationary? So this keeps you informed of your body's location in space. What's your orientation? And it's controlled by gravity. So if you remember when we talked about our auditory system and we had mentioned that there were those semicircular canals, those guys right in the middle that look like a bunch of loops. Those are fluid-filled tubes, and the fluid will react to gravity, and they'll move in a predictable way depending on how we're moving and how gravity is affecting us. So those canals are the biggest part of telling us, are we upright? Are we spinning? Are we stationary? So if you think of, if you ever sit on an office chair and spin around really quickly and then stop, you feel like you're still spinning. And that's because the fluid in those canals is still sloshing around. So it takes a minute because your visual system is telling you you're stationary, but this vestibular system says you're still moving, and you get dizzy because of the disagreement. So this is really important to keep you oriented and balanced. And if it's not working, if you say get an inner ear infection and you're having lots of pressure in your ears, it can make it very likely for you to fall over or pass out because you're getting conflicting information. And as I said, that is our very, very basic covering of those 2 systems. We keep it pretty brief here. I can recommend that with the vestibular and kinesthetic system that you be able to answer a question that might ask you which one does which. Because we talk about them back to back, they're easy to mix up. So I'll leave that hint there. And yeah, we'll come back on Wednesday and visual systems.